You might want to be closer to the screen.
It's all yours. Okay. So hi everybody, my name is Brian. I'll be giving a presentation similar to the one in the analysis class in case you guys were there, but this is an overview of Chapter 1 and Chapter 2 of my eventual master's thesis. So this thesis focuses on using a global trawl dataset that was put together by a group called Fish Globe. And my motivation for using government trawls, which are used to inform stock assessments, is because they have a couple unique features. The total trawl dataset is very long-lived. Governments around the world have been doing trawls for about 50 years to inform how much fishermen can take of a certain species. And because they've been done in so many different places, you have a lot of different environmental conditions that can be met in the survey, and also you have data across national boundaries, which is a big obstacle when analyzing data sets from just one country. You kind of lose effects as you get close to the border. And because this practice is inherently destructive, the stuff that we trawl up dies every time we sample it. I think we have kind of a responsibility to maximize the scientific use of this data, and hopefully it's not being used just to inform industry, but also to maybe glean something from the marine community ecology. So I'll be working with people at Dow, DFO, and Woods Hole. And the data set I'll be talking about covers continental shelves of 8 different nations. All of these areas are highly fished because that's the reason that trawls were conducted there in the first place, to inform how much we can take of the species that are there. And overall in my thesis, I hope to synthesize results from all these different places, mostly in the North Atlantic, North Pacific, and the Arctic. Append the data set where possible. There's in particular one region in Canada that doesn't report its data to the rest of the country for pretty much no reason, and it's Newfoundland. So I'm trying to get DFO Newfoundland to allow us to use their data set. Because every other DFO region does.
Good luck.
Yeah. So then in Chapter 1, I'd like to write functions that I can then apply to each survey region, and as this project hopefully gets updated in the future, you can just use the functions I write with new surveys. Analyzing trends in biodiversity, stability, and ultimately getting like resilience figures for these places. I hope to associate any trends I see with climate and harvesting pressures, and that's going to be key in the study of, usually marine community ecology focuses on like natural areas, like less disturbed areas, but all of these places are highly fished, so that has to be kept in mind at all times. So the applications for this thesis is to identify regions of high stability, in particular in northern communities. I was interested in this project in the beginning because I wanted to see if the latitudinal biodiversity gradient that we often hear about in class, that like as you go north in the northern hemisphere, you should have reduced biodiversity. That can break down as climate change changes the thermal patterns of the world. Then in finding like regions of high stability, so less turnover of species and maintenance of biodiversity over time despite harvesting and despite climate change, hopefully we can find some mechanisms for that resilience. And then use those mechanisms for resilience to inform ecosystem managers who are seeing this shift from cold water species to warmer water species, both in the northern hemisphere and south. So this is the data set. It's called Fish Globe, Fish Biodiversity Under Global Change. It's created by this consortium of scientists, and it was once a UN Decade project. I think that's how I got the money to get off the ground and put together all these surveys, but it has since run out of that money, or the decade is over. So they have been applying for funding. There's a conference coming up in like next week to basically determine if this lives beyond this year. And it would be a real shame for it to go because it's like, I'll show you the size of it in a second. The goals for the survey were to standardize the metadata for all these trawl surveys because every country has their own unique standard practice, but a lot of them do stick to the same protocol every single time they sample, so that's helpful. Harmonize all these trawl surveys, put it in one spot, and create an international community of practice. So the 2 chapters would be the thing I've started so far is looking at the Scotian shelf, and that's just one region of all the different colored regions in the data set. I use the publicly available data set that's on GitHub that anybody can access. And that was a big point for the creators of this, is that the the size of this data set, which is about 2.8 million lines. That's like 30% of all the trials conducted ever, but most of them are restricted to not open access, which is unfortunate. And then in chapter 2, all the stuff that I figured out in chapter 1, I'd like to apply to chapter 2 using the functions I wrote in R to then apply to new survey regions. So just on the Scotian shelf, there's about 145,000 entries, which correspond to the observation of a single species during a trawl. So then the data is indexed by the trawl, which is about a half-kilometer sampling effort, but also by species. And there's 42 different variables measured, mostly environmental and biological variables, with a total of 377 unique species observed just in this region. So these are most of the variables at hand. The only important things to keep in mind are like in my dependent variables, the stuff I'm looking at. It's biological variables about the species at hand, and independent things are what I have access to draw inferences, to make predictions. And that's things like location stuff, time stuff, gear, and temperature. So the SES region, luckily it's one of the most standardized. DFO conducts the surveys, and they had some little variation like when they were starting to do them, but ever since then they keep to a very standard protocol. The gear they use has only changed 3 times, and each time they record what happened when they made that gear change. They don't apply a conversion factor, I think, but they do say what you can see as a result of that gear change. And luckily somebody on my committee has some kind of inside knowledge about how that shaped out. In the beginning, they caught a lot of stuff and then just didn't attempt to identify it and only looked at the species that they were interested for fisheries and then threw all the dead stuff back in the water. So,
what's SCS?
Scotian shelf. So one thing to keep in mind with this project is that sampling effort has changed a lot over time, so that's going to influence how I can interpret results. Especially during years where they're really worried about the fish stock, they sample a lot more to find evidence of, or to better understand like how much they have available. But that's really changed over time. So far I've analyzed these 4 questions in that analysis class. And I'm interested in how biodiversity has changed by latitude, how animal condition has changed over time. That's pretty important for fisheries because the overall abundance of fish doesn't really matter unless you know how much its weight is, how much you're going to get out of that. Where diversity has changed, I'd like to know if sea surface or sea bottom temperature better predicts diversity. Most of the species in here are demersal fish, so you see the result that might make sense. And I'd like to know if richness varies by season, and if that seasonal pattern depends on latitude. So the first question, it looks like over the entire 50-year data set, latitude and richness aren't visually related. But there's a couple interesting things. So there seems to be a peak in richness at like a certain latitude, and I'm going to investigate if that is a hotspot of diversity for some reason. If there's a reason why at 43 you see the highest richness of the data set. But overall, we still get significant relationships between latitude and richness, but it explains really little of the variation, which might make sense because this is across 50 years in a whole lot of different locations. So it's likely that with all these entries, I'm not going to get very high adjusted R-squareds. But it's also worth noting that like recently, the effect seems bigger, which is kind of interesting. So if you're restricted to 1 year instead of the whole 50-year data set, the effect of latitude is more pronounced. And that could make sense as like inter-year variability in climate means that the effect of latitude is getting masked between years. Moving on to animal condition, this is the average animal body weight over time, and it looks like it's decreasing over time. Body weight overall is distributed logarithmically, kind of according to the line tree, so there's a lot of stuff that weighs just a little. Using that log length function for an appropriate distribution of body weight, we find that year is negatively associated with body weight, and species on average are losing about 28 grams a year. So then for this region, as well as for all the other regions I'll eventually analyze, I wanted to take like the dominant species, the ones that we care about the most, and figure out a better model of their body weight that includes more factors in the data set. So this is cod, for example, and for something like a notable decrease in like the 90s or 2000s, I'd like to associate that with historical observations of the catch at the time. So where they caught less, that might be related to the body weight patterns that we see. So then sea surface and sea bottom temperature are distributed like this, and it's funny that this is unimodal and that's bimodal, but that might just come down to sea surface temperature is more susceptible to seasonality. Sea bottom temperature is probably more constant throughout the year. So across the whole 50-year data set, you get this unimodal versus bimodal distribution. And as one might think, the sea bottom temperature better predicts hull richness when compared to sea surface temperature, because most of these species are living near the bottom as it's a draw circle. And one thing that I'll have to check is if these relationships that I find are actually significant or just statistically significant. Because of how many data points I have, it's easy to overanalyze noise. And so if this is not explaining much of the variation in hull richness, sea surface temperature might be a statistically significant predictor, but functionally not really. So then biodiversity does vary by season. These are unstandardized plots for sampling effort. So the total number of species observed in a year is the first one. And then in each of the other categories is by sampling season. And it looks like summer is kind of driving the trend. The most number of things is observed in the summer. And it also shows off the weird sampling practices. Fall was only sampled during a small period in time. So I have data from that time, but it's odd to compare it to the other seasons that were sampled so much longer. And then also sampling season doesn't mean the same thing as calendar quarter, I found out. So even if it was a fall conducted survey, that might not mean it happens in the fall. And so using quarter instead of season, I find that there are significant seasonal effects, and that summer overall has higher diversity, fall has slightly higher. Interestingly, latitude is positively associated with the diversity. So as you go farther north on the Scotian shelf, you get more diverse hulls. And that could be because of a complicated temperature landscape around here, or it might not be that going north is the assumed warmer direction at all times. And then the effect of latitude seems to be reduced during the warmer months. So if there is that breakup in diversity by latitude, possibly because the warmer months break down some biogeographical barrier, that effect is lessened. This complicated plot is if you stepped into each quarter of the calendar, you would watch it change over time in hull richness. And so there's not much of visual patterns here, but maybe in quarter 3 there might be a slight upward trend, quarter 4 an upward trend over time. So in just, I think it's winter, spring, fall, winter, spring, summer, fall. So in summer and fall there's a slight upwards trend over time. So these are some takeaways from the chapter 1 analysis I've done so far, and I'll have to see if these effects are reasonably important or if they're just statistically significant. So now that I've done that, I'd like to clarify the temperature landscape of the Scotian shelf and figure out the relationships between measured latitude, longitude, and sea bottom temperature in particular, because the expectation I might have had coming in that going farther north would result in lower diversity seems to not be true. Then, so far I've been doing all this analysis on hulls, which have become my sampling unit, but over time hulls are not conducted in the exact same spot. And so if you want to make some inferences about how a plot in space is changing over time, you kind of have to make your own boxes and associate bin hulls into some box that they fit inside to say that that's a community. So that's my next plan, is to say that these boxes of longitude and latitude that I set are now communities, and analyzing that over time instead of hulls, because hulls don't happen in the exact spot every year. Then with those new communities, I'd like to study their stability, diversity, and functional diversity over time. There's a package developed by somebody on my committee to study community dynamics using matrix algebra, and hopefully that'll help with that. And then with those different factors, I'd like to get rates of change in all those categories and find subregions within the Scotian shelf in Chapter 1 and subregions within the whole data set in Chapter 2 that demonstrate high resilience, that have low change in diversity and function. So this is the conference coming up, and somebody on my committee, Nancy Shackle from BFO, will be presenting my topic at the conference in France. And at the very end of it, they will be deciding if there's other ways to get money to support the group and hopefully add new trawls to the data set, because it's 50 years long in 8 different countries, but they haven't added data since 2020. So if this thing isn't updated anymore, it'll be a nice historical record, but it will not be a functional piece of new science. So I really hope they find some way to get money to keep this going. It's an overview again of the Chapter 2 sites, and I hope to apply all the same methods from before in all these new places. And that's the committee.